Well, good morning, everyone, and welcome to our last fall Edward A. Ames Seminar. We're putting together a new schedule, and if you have any suggestions of topics you might be interested in, let me know after the seminar. We've got a couple that we think are already planned, but if you have any ideas of speakers, we're always interested to hear your views. I'm Dennis Sikowski. I'm the science director here. And we have a great speaker, Eric Schultz, today from the University of Connecticut. Some of you may know Eric. I feel privileged that I've known Eric now for a number of years, and he's, I'll call him a fish ecologist. And at least I consider him a fish ecologist. And he not only, what's nice from our standpoint is he not only enjoys working with fish, he enjoys working with Hudson River fish, and has some interesting projects in the past. And a few years ago, he came to us with a project as we were contemplating the demise or at least the decline in certain key species in the river, like American shad. Eric reminded us that the fish in the river interact with one another, and who eats who is very important in the river. And as the river has changed over the years, particularly in light of things like invasion of the zebra mussel and presumably climate change, that not only have we experienced changes in the river, but we're expecting changes in the river to continue. And should we expect the same sorts of interactions and same kinds of fish and their quantities in the river is I guess, an open question, and something that we would like to continue to pursue and learn more about it. So Eric not only proposed a very interesting project to take a look at who's eating who in the river, but to me, the greatest challenge in this project was actually getting his samples to do it, and which would probably be a lecture all by itself, but I admire Eric's extreme patience in trying to get samples out of the utilities and their consultants, which some of you that may have worked with data in the past know how difficult that may be, but he succeeded in doing that, and that is at least worth commendation. But we're going to hear about some of this work, and so I'm pleased to introduce Dr. Eric Schultz.
Thank you, Dennis. Yes, that is, the story of getting the samples is a long one, and I'm not going to go there because to give the story any justice would take more time than it's worth, perhaps. But if you want to talk about it afterwards, go ahead. So I want to start out by thanking my two co-authors and describing it a little bit. The first being Mike Smearsich. And Mike did his master's thesis in this project. So the project supported him to do his master's research. Mike graduated from the University of New Haven, although he hails from New Jersey. And he's back in New Jersey now teaching high school at Seton Hall Prep. But he's an inveterate fisherman, and he liked the idea of working on Hudson River fish ecology. And then my other co-author is Dave Strayer, who's likely to be known to many of you. He recently retired as a senior biologist at the Carey Institute for Ecosystem Studies. And David's role in this project is some of his previous research was the inspiration for this work. He provided some of the environmental data that you will see proved instrumental in figuring out some of the story. And he remains an important collaborator because my data analyses in this rather rich data set continue. Now that Mike's done with it and is down teaching high school, I'm finding that I'm still delving. So data's been important in keeping that going. So, all right, Baby Food Chronicles. So I came up with the title Baby Food Chronicles really just thinking, well, what's a kind of semi-humorous way of referring to what we're doing here? And it turns out that there really is a Baby Food Chronicles blog that I found on the web, even after coming up with a name. And clearly, a youngster here is fairly interested in the concept of baby food chronicles, so got me to thinking what's interesting about this. So some of the take-homes that I'd like you to come away with are that this project, with the help of the Hudson River Foundation and the help of the long-term monitoring program, represents a rather a data set on feeding ecology of early stage fishes that is unprecedented in its length. So we're going to talk a little bit in the first part of the talk about long-term data sets and their value and how that plays into determining what's going on with early stage fishes and estuaries and why that's important. So that'll be the first part of the introduction to the talk. Now, the second interesting component of this project is that it's what one might call retrospective descriptive ecology, meaning we're not doing experiments here. Instead, we have a data set that frankly was collected by, in some respects, by other people or designed by other people. And I came to that long-term sample series with an a priori hypothesis. That's good. It's good to do hypothesis-based science rather than simply describe it. But then a kind of interesting feature of our analyses, in addition to having an a priori hypothesis, there were other kinds of information we could gather so we could test for the importance of our a priori hypothesis relative to other intervening factors. So those are two kind of major components of the project. So first, what's interesting about long-term studies and what's interesting about the feeding ecology of early stage fishes and estuaries? Well, long-term series of samples give you the opportunity to assess the impact of the unexpected. So it's in regular ecology, long-term data sets aren't that common. There are some long-term ecological reserves, such as the Hubbard Brook Experimental Forest. And in fishery science, because there is an industry that's paying for the samples, and there are federal agencies that need to gather long-term data, there are some long-term data sets. In other kinds of ecology, there really aren't too many. And we might ask why they're worth taking the trouble to collect them. So we know that we have a changing world, and many times we're in the position of reacting to the changes and trying to figure out what happened once the change occurred. If you have a long-term series of samples, then you can make a much better, more precise and accurate judgment of what the effect of that unexpected change was. So that may assist us in managing these systems in the face of changes that have already occurred, or perhaps even in anticipating the changes that we haven't yet seen. Now, I also note here that long-term studies are great opportunities to do collaborative studies, because many times we're bringing multiple kinds of measurements and analyses to bear on these sorts of situations so that you can have great collaborations. So we also have, in addition to long-term ecological studies, monitoring programs such as the one that we made use of in this project. So in that case, industry is charged with monitoring a system like the Hudson River, and if it's a well-designed kind of monitoring program, then that gives you the opportunity to actually look at kinds of changes that may not have been anticipated when the monitoring program was actually designed. So the monitoring program was designed to evaluate the impact of an industry, in this case, power plants, and the water that they withdraw. But I believe that we can make use of these studies, of these monitoring programs to look at other things. All right, so why estuaries? Well, estuaries are a place where the unexpected occurs on a regular basis. So they're dynamic systems. On a short time scale, we have tidal changes. On a longer time scale, we have things like changes in, for instance, human habitation. So estuaries are strongly impacted anthropogenically. We have many people that tend to live in estuaries where freshwater systems like rivers meet the sea. And so anthropogenic impacts occur on a regular basis. I said invasive species are a really good example of a kind of perturbation that occurs frequently in estuaries because of shipping traffic that brings these invasive species from other parts of the world. So when I say that monitoring programs might enable us to assess the impact of the unexpected, certainly one thing we might be able to do is look at the effect of an invasive species. Now, estuaries are important places to look at impacts on fish, so we might be interested in looking at how these unexpected events affect species of interest, perhaps commercial interest, among the fish fauna. And estuaries indeed are important to the fish fauna. Estuaries represent nursery habitats for many fish species. So with some effort in the eastern United States, you can collect 100 species in an estuary typically. Now, some of those are incidental, whereas some of them live there on a regular basis. But even those species that you can collect on a regular basis in an estuary typically do not live for their entire life cycle in an estuary. Instead, it's most common for a species that spends most of its life cycle out in coastal waters to come into estuaries to spawn. And the larvae concentrate in estuaries. Estuaries are very productive places where there are lots of feeding opportunities, and so growth tends to be fairly rapid. And then at an appropriate time, those early life stage fishes withdraw out to the coast. So this so-called nursery habitat, because most of the mortality occurs in the early stages, the estuaries are places where what we call year class strength is determined, meaning a quantity of interest to us, which is how many fish are going to be out there for us to exploit, to eat. The events that determine when that year class strength is, what that year class strength is, tend to occur in estuaries in the early life stages. So we'll be interested, we in general are interested in the impact of unexpected events in estuaries and how those are affecting the population dynamics of fishes. All right, so as I said, baby fish grow fast in an estuary. Let's take a quick look at the food web. This is actually a representation of a Hudson River food web. We have 3 species of interest here, the striped bass, the American shad, and the bluefish. I'll be talking about the first 2 of those. And both of these species, when they're adults, tend to eat fish, in the case of striped bass, or large invertebrates, as is the case with shad. The larger juveniles tend to eat large invertebrates. What we need to see from this representation of the food web is that there is a pelagic component to the food web here and a more benthic component here. Phytoplankton, for some reason, are really represented just as part of the benthos, but it really represents primary production, photosynthesis, that occurs in the pelagic zone that may settle out to the benthos. So in the benthos, near the bottom, we have production that is going through benthic organisms such as clams, such as crabs, that feed benthic fishes. Some of that benthic production goes up into the pelagic zone. The pelagic zone has invertebrates like copepods, zooplankton like copepods or larval crabs, and baby striped bass and baby shad rely heavily on that production in the pelagic zone and in some respects to the benthic production as well. So that rapid production that occurs in the Hudson River enables these young fish to grow rapidly. So in a more kind of schematic basis, what we have here is we have the phytoplankton production and some interaction with benthic production that might be occurring with of attached vegetation, or what's called periphyton that's growing on the bottom. And that production supports what we call consumers. So those are herbivores that might live on the bottom, such as would be the case with this organism, which is an amphipod called Gammarus. or a pelagic zooplankton such as that represented here, the Cladospora and Daphnia, and these organisms are preyed upon by these baby fish. Okay, I'm not going to talk long about the Hudson River estuary because you all are expert in this estuary to an extent more than myself, I am sure. However, I should say, for the sake of putting this talk in context, that the Hudson River estuary is a very well-studied system. It's been monitored extensively, particularly by both the state and by the Hudson River utilities since the early 1970s. And these long-term monitoring programs that have stretched the length of the river from the dam up in Albany down to the mouth of the river where we are now have proven useful in seeing the effect of environmental changes that the estuary has endured. Now, I've belabored several times that the estuary is subject to unexpected events, and this certainly was an unexpected event of a rather great magnitude. This attractive little organism, this bivalve, has proved to be a scourge in North America, introduced in ballast water by the shipping industry up in this portion of North America near Detroit, Michigan, and came from Eastern Europe and Northern Europe and spread rapidly both across the zebra mussel and its congener, the quagga mussel, spread throughout fresh waters of the United States out to the west and down to the south, and spread rather completely through the Hudson River Valley. Now, one of its effects is represented here graphically with this shopping cart. It spreads across the bottom. It's represented a real problem for industries because it clogs intake pipes, among other things. Some of the other ecological effects is that it does increase the amount of hard benthic habitat as it spreads, so it can be habitat for other organisms. And for the purposes of my talk, an extremely important thing about it is that because it is so numerous and because it filters a great deal of water, it tends to, as we will see, clear the pelagic primary production associated with phytoplankton from the water column. So it clears the water column. One last important thing to mention about it is a bit of natural history. Unusually for a freshwater bivalve, the zebra mussel has a larval stage that is planktonic and free-living. So most other freshwater bivalves, the freshwater mussels living in our waters, have a free-living stage that attaches to fish. But this organism produces a veliger larva that swims around for a couple weeks and then settles. So that assists in its spread, but it also has some other implications for feeding ecology, as we will see. Okay, David Strayer and his colleagues at the Carey Institute documented the ecological effects of the zebra mussel in a series of papers. This represents schematically the kinds of changes that were associated with the spread, the invasion of the zebra mussel in the Hudson River in the early 90s. So in several studies that were published in the late 90s, the early 00s, and even since, they documented that some of the changes occurring to the river were in the negative realm. So a negative change, for instance, in the phytoplankton, as I alluded to earlier, as the zebra mussels clear the water, and a positive change to some parameters as well. So, for instance, littoral fish benefited from the invasion of the zebra mussel. A littoral fish is a fish that lives in shoreline areas or in shallow waters, and because the zebra mussels cleared the waters, more sunlight reached the near shore bottom, which increased the amount of production associated with attached plants, increased the amount of habitat for these littoral fishes, and seemed to have increased the abundance and growth rate of those littoral fish. Pelagic fish particularly the early stages of fish of, for instance, striped bass and shad, as we will see, experience negative effects at various times. And I'm going to skip over a lot of these and simply note that, for instance, phytoplankton, as we'll see, the number they gave that was a nearly complete loss of the green organisms in the water column that represent the base of the food chain for those pelagic fishes. Okay, some of David's work documenting the impact on early stage fishes were the inspiration for the study that I'll be describing to you today. So what we have here is a plot of the estimated change in the growth rate of early stage fishes, according to several different studies of a variety of species. So what's in the parentheses here represent different sampling programs. Long-term sampling programs that might be done have been done, for instance, by the Department of Environmental Conservation of the state, or might have been part of a study associated with the Hudson River Utilities. LRS stands for the Longitudinal River Survey. It's conducted by Norman Doe Associates, supported by the Hudson River Utilities. So these are a variety of different studies, as I said, of a variety of different species, broken out into open water pelagic species and then littoral species. And the Strayer and his colleagues attempted to estimate the change in growth rate that was in comparing times before the zebra mussel arrived to after the zebra mussel arrived. So looking at long-term trends, what appears to have been the change in growth rate. And let's, oops, sorry, I hit that button prematurely. I want to just direct your attention to striped bass here. So this was an effort to determine, based on change in size distribution over the season, so looking at the shift over time within a season in the modal size of early stage fish. So if that modal size, if the size distribution has gotten bigger by a couple millimeters a week, for instance, in early years, before the zebra mussels arrive, and then after the zebra mussels arrive, maybe that size distribution only shifts by a millimeter a week, for instance. Just drawing numbers out of my hat here. But that was the basis for concluding that the striped bass growth rate may well have been negatively impacted by the zebra mussel's arrival in the Hudson River, suggesting that that collapse of the pelagic production, the loss of phytoplankton, may have affected the food supply for striped bass. Other species seem to have been affected not so much by growth rates as by abundance. So these graphs are set up in much the same way as before, with the different points in the x-axis representing perhaps different species or different sampling programs. The y-axis in this case is a before versus after comparison of abundance. And I'm going to draw your attention to the plot for American shad. And this graph suggests that the abundance of American shad larvae, baby shad, has decreased as a result of the zebra mussel's arrival. So when David published these studies, I thought, that's very interesting. It seems to be a compelling story. It would be good to identify the mechanism associated with these decreases in growth rate and with this change in abundance. If the explanation for these changes is correct, then we should be able to see a change in the feeding ecology of these species. We should be able to see that their diet has changed and that their feeding success has decreased as a result of the zebra mussel invasion. And we have a long-term sampling series to demonstrate that. So going back to the schematic that we had before, we have here, prior to the zebra mussel's arrival, a certain amount of production in the benthos of the pelagic zone, supporting consumers that go to fish. Now we have the appearance this figure, large bivalves, would just say that's the zebra mussel increasing in abundance, and what that's doing is it's decreasing the amount of production in the pelagic zone. It would therefore represent potentially a competitor for food of these edible consumers, which is then going to have an impact on fish. So that's the setup schematic of the explanation for what might be going on. Now, in this long-term sampling series where we have opportunities to examine the feeding ecology of fish that were collected even before the zebra mussel arrived, we also have a number of other environmental variables that we can test for their influence on responses of interest. So the long-term sampling series also has data on temperature, dissolved oxygen, the discharge and flow in the river, and salinity. This is important because the estuary is undergoing many changes, not just that of the zebra mussel invasion. So it's going to be important to be able to employ these other potential explanatory variables in investigating the effect of the zebra mussel. We also have, by way of quantifying what's going on, these important features of the feeding ecology, chlorophyll. So that's a representation of the amount of phytoplankton or pelagic production, the abundance of different kinds of prey, ideally both in the water column and on the benthos, and then some estimate of the abundance or the feeding activity of that zebra mussel. So that's kind of the setup for how we went about our work. The objectives for our project was to characterize the diet composition of early stage fishes in the Hudson River over a multi-decade time scale. And then the idea there was to assess the extent to which the zebra mussels may have affected this feeding ecology in combination potentially with other ecological factors that may have influenced the feeding success of these early stage fishes. So as I've alluded to earlier, the survey that provided the samples for this work is called the Longitudinal River Survey. That was conducted, paid for by the Hudson River Utilities and conducted by Norman Doe Associates. That sampling program was what's called a stratified survey. So the survey design, which has been conducted in substantially similar fashion since the mid-70s, divided up the portion of the river that we call the estuary between the dam down to the mouth into 13 different regions. So every week or every 2 weeks, depending on the time in the year, there is sampling occurring in every region of the river, giving us an opportunity to look at what is going on all the way along the estuary. So sampling in every part of the river, sampling over an entire season, sampling every year. The sampling, in addition to the environmental variables that I just mentioned, includes sampling for young fish. So they drag a net through the water and collect baby fish. And thank goodness, they actually saved those samples. So those samples, some of them have been archived. And so the Norman Doe Associates maintains a big warehouse that has samples of fish going back to the early 90s. There was also a sample of fish that was collected in 1998 that was transferred from Norman Doe Associates back to the New York State Museum. So that was an important source of evidence for what the estuary was like before the zebra mussels arrived. One other thing to mention about these samples is they are taken at night, or at least when the sun sets. So they're archived, they're saved in formalin, and we were able to, as Dennis alluded to, with some effort, get access to these samples. We then took them to the University of Connecticut and we analyzed them. Now, as I said, they're sampled every year. We did not analyze every year of fish samples in our study. Instead, we subsampled, and we subsampled in 3 different time segments. So based on work that was done at the Carey Institute on the effect of zebra mussels, we felt there was a time period that was before invasion, This is not actually before the zebra mussels arrived in the river, but before the zebra mussels had a strong detectable effect. So this was what we call the pre-phase. Then there was the impact phase, and this is when the invasion had its sort of peak effect in terms of clearing the water column. Now, since then, as some of you know, there has been kind of a recovery phase. The zebra mussels are still in the river. They're still quite abundant, but some of their ecological effects have been moderated somewhat. That was actually a nice thing about this study, is that we're able to perhaps bookend this strong ecological effect by, one, the time before they arrive, and two, a time when the system may be recovering somewhat, which might enable us to sort out the effect of the zebra mussels from other long-term changes that the estuary is undergoing. So these are the years that we subsampled. We had 3 years from the pre-period, including that year that of samples we retrieved from the New York State Museum, 5 years within the mussel impact phase, and then 7 years of recovery. One other feature about our sampling design is that for at least one of the 2 study species that we focused on, we also had the opportunity to take baby fish from the upriver and downriver portion of the estuary. This is important because this upriver portion of the estuary is where the zebra mussels are most abundant. Zebra mussels do not like salt, and so this portion of the river depicts where they have their strongest ecological effect because they're most abundant. Downriver, there really aren't very many zebra mussels. We expected that their ecological effect would be muted. So because we were able to get both upriver and downriver samples of some of our fish species over the entire time period, this represented an additional opportunity to perhaps sort out the extent to which mussels were affecting the feeding ecology of fish relative to other ecological impacts. Okay, let's talk a little bit about how we quantified some of our responses. So as I alluded to, one of the things we're interested in is how zebra mussels may have affected the diet composition, the things that our baby fish are eating. In order to quantify that diet composition, we use something called the prey-specific index of relative importance. I'm not going to completely walk through this formula, but I will tell you that it's a weighted index in which we quantify both the number of a particular prey and the weight of a particular prey in determining its importance to the diet. So a prey item might be important because it's very numerous or because it is fairly heavy. So it's 2 indices of importance to the diet that are combined in this index. And this represents some of the organisms that we see. So here's a copepod, here's an amphipod, and here's, for instance, a Daphnia podacea. Okay, another thing that we did in quantifying what's going on with the feeding ecology is we're very interested in feeding success. As I alluded to earlier, we were interested in whether the appearance of the zebra mussel would reduce the feeding success of baby fish because there's less food around for them. So we have 2 different quantities representing feeding success. One of these is a short-term index of feeding success. How much do these baby fish have in their tummies when they were collected? And so in order to quantify that, we took the stomach contents out and we settled them in a little centrifuge tube. And we estimated the volume of the settled gut contents. Now, that volume of settled gut contents is going to be very dependent on the length of that baby fish, as you might imagine. Bigger fish have more stuff in their stomachs, as we see here in this graph. So overall, you can see that there's a trend towards larger fishes having more stuff in their stomachs. So if we're going to look at interannual variability in feeding success that might be the result of the zebra mussels, we have to take out the effect of length. So in order to do that, we look at, let's say, a size-specific volume of gut contents by looking at the difference between the overall relationship between gut volume of gut contents and length and an observed point. So the fish up here above the line have relatively full tummies, and the fish down below the line have relatively empty tummies. And we're asking whether the zebra mussel invasion is shifting the overall position of that relationship. Okay, another index of feeding success that's important for our study is a more long-term measure, which is essentially how much meat do these fish have on their bones. There's a term called condition that people that work with fishes look at, which is the amount of weight per unit length. So what we did with our fish is after dissecting out their gut contents, we dried them in the drying oven. So we look at their dry mass. Now, of course, a longer fish is going to be heavier. So just as was the case with the volume of gut contents, we have to take out the effective length in looking at the amount of dry weight. So here again, we see a plot of dry mass against length, and you see some scatter around that relationship. Individual fish that are above that line are in what we call good condition. They've got relatively much meat on their bones, more protein, probably not more fat. These fish are too young to have much fat on them. So they've probably got more musculature essentially. What does that mean in terms of their feeding success? That means that over the last days to maybe up to a week for a young fish like this, their feeding conditions have been good. Whereas fish that are down below the line, their feeding success has been poor. So we were interested in whether the zebra mussels and other environmental factors may have affected the position of fishes in that overall relationship. Okay, environmental variables that we were able to employ in our analyses include some abiotic factors. As I mentioned earlier, some of these were collected as part of the longitudinal river survey, so we could look at the effect of dissolved oxygen on feeding success. Salinity and temperature. All of these were collected pretty much in the same date and at the same location as the baby fish samples that we analyzed. Then there's a variable called river discharge, which is essentially the amount of water that's coming down the river. Those data are collected by the USGS on a daily basis, and we average them over the week that the fish samples were taken. Then there are biotic variables. Now, these biotic variables were collected by the Carey Institute. That included the abundance of amphipods on the bottom for the years of interest, the amount of chlorophyll in the water column, the abundance of copepods, and, importantly, the amount of filtration that the zebra mussels in the river were exercising, as it were, a combination of their abundance and their biomass. Now, these data were collected at Kingston, and they were collected on a different schedule than the ichthyoplankton samples. So rather than trying to align them with our samples, we took annual means of these biotic variables and asked essentially statistically how much those were able to predict the feeding success of our organisms. Okay, now those biotic variables have some relationships that I've mentioned already. So here we have a plot of, on this axis, the amount of chlorophyll A in the water column, and on this axis, the zebra mussel feeding rate from the before zebra mussel period, when the mussel feeding rate was near zero. Actually, that is zero, see from this axis. So there were no zebra mussels to speak of in the early part of the river. And then upon the invasion, that feeding rate shot up. And here's the peak mussel invasion period till about 2004. And since then, it's moderated a little bit. Let's look at what happened to chlorophyll. So chlorophyll was high before the zebra mussels arrived. And then, as I've mentioned, it pretty much collapsed. There have been some years in which chlorophyll has returned to the water column, and it's easing up slowly, but this demonstrates the strong effect that zebra mussels had, looking at year-to-year variability on the amount of phytoplankton in the water column. Now, I had expected that the zebra mussel invasion would also show a sharp effect on zooplankton. As I mentioned earlier, that had been the suggestion. However, we are not seeing that in the copepod data set. So the y-axis of this figure is mussel feeding rate as before. This axis is the abundance of copepods, adult copepods, in the water column on an interannual basis. And this is a correlation analysis, which suggests that there is no significant relationship between these. So when you're looking just at the copepods, that reduction in the amount of phytoplankton in the water column did not appear to reduce the abundance of an important food source for baby fish in the water column. I only really discovered this in the course of doing this project. It's a little bit unexpected. Benthic production is also, according at least to the abundance of amphipods, as depicted here, and there's a little amphipod, was also not affected by one way or the other by the zebra mussel invasion. So the abundance of amphipods has remained relatively, it's highly variable, but it hasn't either collapsed or increased. It was fairly low before the zebra mussels arrived. It seems to have spiked right around the time that zebra mussels arrived, and since then it's kind of wandered around at a fairly low level. There are breaks in this data series. Dave Strayer tells me that these data are extremely hard to collect, and knowing what he knows now, I think they would have monitored amphipods in a different way. But it is an important source of information for us on the amount of production of, let's say, fish prey in the benthos. Okay, I'm going to talk about two species here, and the first species that I want to talk about is the striped bass, and I'm going to briefly go through the natural history of striped bass. This was an important fishery in the Hudson River estuary. It is a large, can be a large fish. It's a generalist predator that is mostly anadromous, meaning it goes up into rivers to spawn. There are some non-migratory striped bass in the Hudson River as well. It certainly is an ecologically important fish, and at least until the 1970s, there was a large commercial fishery for it. So there are lots of baby striped bass in the river. We were interested in the extent to which it affects the feeding ecology of these baby striped bass. One of our predictions was that diet composition would shift to benthic prey during years of the zebra mussel impact, especially upriver where the zebra mussels are abundant. So a little bit on the methods here. We had 30 fish for every year per location, both upriver and downriver, and then we quantified the prey-specific index and relative importance that I described to you earlier. Okay, so here's some data on the diet composition of these fish. It's rather similar upriver and downriver. So the blue bars represent the upriver diet composition, the green bars downriver. And I've broken out, although we had 15 different sorts of prey categories here, I'm showing you the four most important ones. And copepods, amphipods are important in both of those. Those are the two prey items that are of overwhelming importance, and they're about equally important upriver and downriver. However, mysids, which is a primarily marine kind of invertebrate, was more important downriver than upriver, and conversely, Leptodora, which is a rather peculiar skeletal-looking kind of cladoceran, tends to be a freshwater cladoceran and is more important upriver than downriver. Okay, we have here a plot that is called an ordination plot. It attempts to represent the relative change from one year to the next in the entirety of composition of the prey. So it's taking these 15 variables of prey composition and representing them in a two-dimensional plot. Points, that is years, that are close together in this plot are similar in terms of prey composition. Points that are farther apart are different in terms of prey composition. I've represented the different symbols in terms of these four different time periods: pre-invasion, the impact of the invasion, and the recovery. And we predicted that there would be some change in diet composition in these three different time phases. And we don't actually see that. So we see here the early years, for instance, this is 1992, here's 1991, here's 1988. They're kind of all over the map, and especially these 2 points are together with some rather recent years, like 2011. So with respect to these three different time phases, we're not seeing a clear signal of prey composition. Now, I should say that I'm going to actually, an unfinished task, is I'm going to be looking at these data in more detail, particularly with respect to zebra mussel feeding rate, to the extent to which zebra mussel feeding rate actually influences these variables. I'd prefer that we hold questions until the end. That's all right. Now, this analysis here, which is called an analysis of similarity, does tell us that there is significant variability from year to year. It's just that it doesn't seem to be falling out in terms of these 3 different time periods. Now, with respect to feeding success, we had a clear a priori prediction that feeding success should decrease during the period when zebra mussels are feeding actively. And that was based on the previous findings of reduced growth and of reduced abundance. So I'm plotting one result here of many. The y-axis is that representation of long-term feeding success. So this is fish condition. It's adjusted for size, and it's represented for a variety of years. And we see here the condition was relatively good, and then it fell right around the time of the zebra mussel invasion. It wandered down to this slightly low level. It rose, and it fell again. Now, admittedly, I'm drawing lines between these points, which is probably over-representing things in the temporal trend. It does help guide the eye, but I'm not suggesting that there was any kind of smooth change from one year to the next. This is a result of an analysis of covariance that takes that dry mass and seeks to explain it in terms of the length of the fish, the year, and an interaction between year and length. And all of these predictors are highly significant. So for me, what this means is that there's significant interannual variability in feeding success of these fish, and that needs some explanation. So we used a statistical technique called model selection to look at the environmental variables I mentioned earlier, the extent to which they could explain variability in feeding success. So we have the different abiotic variables here and the different biotic variables here, and we threw all possible models at the result and sought those models that fit the data best. Another thing we're interested in doing is seeing the extent to which, once we had identified the best model, how much variability in feeding success it explained. So we sought to quantify the condition at relatively poor conditions, low levels of predictors, and high levels of predictors. So I'm not going to walk through all of this table. I will tell you that we have 5 different models here for both the upriver data set and the downriver data set. Each row represents the predictors that went into the model. So the best model here identified salinity and zebra mussel feeding rate as the predictors affecting feeding success. And there are other variables here that may be familiar to some of you, but I'm not going to belabor here, representing how well the variables actually fit the feeding success data. To summarize, though, I would mention that for upriver, the best model indicated that in times when salinity was slightly higher in this freshwater period, freshwater section of the river, that feeding success was higher, and that mussel filtration rate had negative effects on feeding success, as predicted. In terms of the overall sensitivity of feeding success to these environmental variables, we found that mass at length was about 1/3 higher when salinity was higher and mussel filtration rate was lower. So that's a pretty pronounced effect in terms of mass at length for a baby fish. Downriver, the abundance of copepods and dissolved oxygen were important. More copepods, feeding success was higher, and for reasons we don't fully understand, when dissolved oxygen is slightly lower, we aren't talking about hypoxia here, but we are talking about slightly lower dissolved oxygen, feeding success was higher. Okay, so conclusions for our striped bass analyses were that copepods and amphipods were important prey. We did find significant variability in diet composition among years, but it doesn't at this stage seem to be that clearly mapped on zebra mussel the zebra mussel feeding, but stay tuned, I haven't really done a proper analysis of that part yet. Feeding success varied among years, and it's pretty clear that upriver zebra mussel feeding rate is having a negative effect on striped bass feeding. Okay, another species that we analyzed in this study was American shad. And American shad, as Dennis noted in his introduction, is a species of particular concern here in the river. Few notes on natural history. It is our largest herring. It is another anadromous fish, so it goes up into rivers to spawn. And it has been of great cultural and commercial importance to this coast. In Connecticut, we actually have a shad derby every year. Even when the shad are not that abundant, we go ahead and have the shad derby. And the catch and the stock size of shad has decreased sharply since historic years when there was a lot of dam building, and has continued to decline since then. And the Atlantic States Marine Fisheries Commission and folks in New York have been studying how to enable recovery of the shad. Even in recent years, the spawning stock biomass of shad has continued to collapse. And one of the possible mechanisms that was suggested for the continued decline of shad has been perhaps that maybe there has been a diet shift, and particularly on the early life history stages. So for analysis of shad, we divided life stage for several reasons that I won't really show you here. We decided to divide our analysis into the larval stage and the juvenile stage. We analyzed diet composition as we did for striped bass, so that has changed. That does change with development. For larvae, copepods, and this Cladocera and cold Belsmina are most important. Whereas once the fish get bigger and get to the juvenile stage, they start shifting to feeding on larger items like insects and amphipods. As was the case with striped bass, diet composition varies significantly among years. It doesn't clearly shift with invasion period. Feeding success is an interesting data set, of course, and here we have represented the same way as we did before, the feeding success of larval shad, which was high in years before the zebra mussel arrived, fell sharply, recovered a bit, fell again, recovered a bit, fell again. It's kind of been wobbling around in an intermediate level. Juvenile feeding success, that is their condition, didn't go through a kind of high value and then a collapse. However, it has been through some interesting negative excursions that correspond to the same negative excursions in larval data. So it's pretty clear that years 2003 and 2009 were not so good for baby shad in the river. We did mixed effects modeling of the same variables as we did for striped bass. What we found for larval shad was that the chlorophyll and copepods are important for feeding success. They do better when there's more chlorophyll in the water, when there are more copepods in the water, when the water's a little warmer, and when mussel filtration rate is positive. So they like, after the effects of these variables have been taken into account, they actually like it when zebra mussel feeding is high. Same story with juveniles. Chlorophyll is important, copepods are important. In this case, river discharge was important, which is still sorting out the effect of that. And again, mussel filtration rate. In terms of the sensitivity analysis, these variables combined have a huge impact on feeding condition by factors of magnitude in the case of larvae. Okay, so conclusions for American shad. We found that copepods and Bosmina and amphipods were important prey. We found variability in feeding success among years. And zebra mussels had an effect on feeding success of both life stages. So what happened here? Going back to our earlier schematic, we had predicted that zebra mussels would negatively impact the feeding success of these two early stages of fish, based on previous findings of reductions in growth rate and abundance. So when the zebra mussels arrive, they represent competitors for these early stages of larvae because they are feeding on they represent competitors on the food source for the early stages of larvae. So they're competing for this, with the zooplankton for pelagic production. What we found with the striped bass is that zebra mussel feeding is having a negative effect on their feeding success. If you take out the effect of zebra mussels from the analysis, same data, but you just take out the effect of zebra mussels from the analysis, you find that copepods and chlorophyll contribute to the feeding success of stripers. So if there's more production in the water, then the baby stripers do better. And so zebra mussels are having an effect because they're taking that production out. Okay, with a shad, the story was different. So we found copepods are important, chlorophyll is important, but after the effect of those have been included in the model, we find the mussels still remain important. That was a puzzling result. There is one possible explanation, of many perhaps. Chris Knapp, who is a student of Karin Limburg's, recently documented that baby shad eat zebra mussel veligers. Now, we did not find zebra mussel veligers in our diet composition analysis. I found in one specimen, a juvenile shad about this big, I found its stomach packed with bivalves, but they were above the veliger stage. One possible explanation for this pattern is that, yes, zebra mussels are taking out prey for the conventional prey for young shad, such as copepods, such as bosmina, but the zebra mussels are also contributing prey for these shad by spawning baby zebra mussels. And that we may not have picked this up in our diet composition data because those veligers perhaps do not preserve well in formalin preserved fishes. Okay, so what was so interesting? Well, I hope I've demonstrated the interesting things you can learn from a long-term study of feeding ecology of early stage fish. I would say that these fish have shown a pretty high degree of resilience to a major ecological perturbation. The shad data suggests that the invasive species is both a competitor and a prey for early stage fishes, which I find interesting. One thing that we don't understand, among other puzzling things the data set projects, is we don't understand in the estuary what pathway of production is supporting the zooplankton in fish in lieu of the phytoplankton. So there is bacterial production that is another route of primary production that doesn't go through phytoplankton. It's a very hard thing to quantify. And it's possible that that production is maintaining, for instance, the copepod populations and the amphipod populations, even when zebra mussels are clearing the water column of phytoplankton. Okay, so in terms of thanks, this was a labor-intensive project. We had a large, primarily volunteer workforce picking through stomach contents. We had some, thanks to the Hudson River Foundation, we were able to pay Mike, and we were able to pay some of these students to work for us during the summer. But during the academic year, they did independent study where they helped us analyze samples, and they also learned some additional science. Thanks to the New York State Museum for providing some of the samples for us, and then the National Science Foundation supported the long-term data on environmental factors that I used in some of these analyses. Thank you very much.
Before we take a question, I just want to apologize to the people on the webinar for not welcoming them properly at the beginning of the seminar, and to remind them that they can ask questions as well if they want to write in, I guess, on the right side of their screen. And I apologize to Sarah for not making that announcement as well. So questions.
Yes, Jack.
As you know, I've been running up and down the Hudson for about 8-9 years. I've never seen a zebra mussel and how to grow up, but I do see a smaller mussel quite profusely, and I was just wondering why that was not mentioned in your discussion.
We do not have data on that smaller mussel. I would assume this being in Haverstraw where there's some salt water, it's probably Middleus. And Middleus does not, although it can be abundant, does not have the ecological impact that the zebra mussel had. So they do filter the water, but they have not had the effect on pelagic production that zebra mussels did. It would, of course, be very interesting to look at the extent to which variability in other benthic mussels have had on the feeding ecology of fish. We simply don't have the data for it, unless you know of a data set.
I don't know.
Okay.
Thank you.
Yeah. Yes. Sorry to put you off in terms of asking your question, but I was on a roll, so I didn't want to be interrupted.
I hope I can get it back together.
We can go back to that slide if you'd like.
No, that's all right.
Okay.
There's some inconsistency issues here in terms of one of the, I guess, biggest surprises of what you said was that the zebra mussels did not affect the abundance of copepods or amphipods.
Yes.
So given that,
Yes.
I would be surprised that it would have had any effect at all on the striped bass, since the striped bass are eating mostly copepods and amphipods, so they would have enough food.
Yes.
And the filtration by the mussels shouldn't make a difference as long as you got enough copepods and amphipods there.
Right.
Right. That's one.
Yes.
And then the second, perhaps related, perhaps not, question is, in some of the years where you've had relatively poor condition and feeding success, and it was hard to correlate with the zebra mussel effect, is it possible that reproductive success may have been very high at certain years, so you're having more competition amongst the larvae for the finite amount of food available?
Good. So I've been instructed to repeat or summarize the question for those out in the webinar. So let's take the questions one at a time. First question, were we off base to predict an effect of zebra mussel on feeding success, given that there was no documented effect of zebra mussel on two of the predominant prey, those being copepods and amphipods? The answer is a yes and a no. Yes, in that to an extent that I had not actually anticipated when we undertook the study, zebra mussels were not affecting copepod, adult copepod abundance. Didn't actually expect there to be a positive or negative effect on amphipods. I should say that the early stages of copepods, copepodites, the larval stages, do appear to be impacted by the zebra mussels in the long-term data set. I'm not showing you those results here, but work by Strayer and his colleagues suggests that copepodite abundance is lower, and we are not actually in the diet composition data that I showed you here, not trying to sort out which stage we're looking at. So when we're lumping copepods for the diet composition, we're not making a life stage discrimination. Whereas the environmental data that we're looking at from the Carey Institute, they are, because of the nets they use, just looking at adult copepods. So there might be a bit of a disconnect there in terms of prey abundance. The other thing that might be a bit of a caution is that there is, of course, a difference between standing stock and production for any of these components of the ecosystem. So abundance gives you part of the picture, but what we don't know is how many are moving into that box or out of that box at any one time. So feeding could be very intense on something that is really abundant, in which case you'd find a lot of that prey in the diet. The production could also be even intense when abundance is low. So you could find a lot of a prey item in the diet even when abundance is low. So it's really important to use environmental variables as predictors in looking at diet composition in addition to the the other environmental predictors. So I guess I would caution that when we're looking at rates and standing crop, that we want to be a little careful in looking at long-term patterns and trying to impute exactly what process is leading to that. Now, the second part of your question, as I remember it, is, oh dear, now I've painted myself into a box. Remind me, sorry.
Density, because of
density dependent. Yes. Yes. So you were asking about density dependent effects. That it could be that, for instance, competition among, I think this is what you're asking, could competition among larvae be very high? And so perhaps, this is a testable question, if the abundance of larvae is really high, could that depress feeding success? Not because of other environmental factors, but just because the adults loaded in a whole bunch of babies. We are looking at that. I don't have a conclusion on it yet. So far, we've seen no indications that abundance have reduced feeding success, nor do I yet have an indication, I'm not done with that analysis, that a year of high feeding success would also have led to a strong year class, meaning a lot of larvae coming out at the other end. That's on my to-do list right now. Okay?
I want to think of, we got a bunch of.
Oh, yes. You want to read them, Sarah? Thank you.
I'll speak from here because this microphone should work.
Okay.
So, David Barnett asks, you referenced river discharge as a variable.
Yes.
Do you have any sense of the effects of upstream wet and dry years and severe events such as Irene and Lee?
So we don't at this point have the, we've not done any what we call event-based analysis, where we look at the effect of these tropical storms and a real pulse of discharge. I haven't yet taken a look at kind of within-season variability and parsing that out from among-season variability. We did find that discharge was a predictor on feeding success for larval shad, juvenile shad. I'm interested in that. It actually is a bit counter to what I would have expected based on previous research done on the success of larval stage shad in the Connecticut River. Years of high discharge are not good for year class success. So I thought it was very interesting that we found a positive effect of river discharge on feeding success, but I haven't sorted out why that is yet. So I hope that represents a complete enough answer to this question.
Do you want to try Dr. Walden's question there?
I do.
The one you got highlighted.
Okay. This is Hoffman, actually. Is the one.
Okay, sorry.
Well, I'll begin with him first. How can you separate the effects of zebra mussel invasion from the increase in striped bass population due to fishing restrictions?
Right. So the way I would do that would be to look at the abundance or standing stock of larvae in the river as another predictor. And as I said earlier, we, at least in our current analysis regime, I've not found any effect of striped bass larval abundance on feeding. But this is still work that's ongoing, so I do need to pursue that in more detail. But I think that would be the way to go about it, would be to, because these year class, these monitoring programs do have an index representing the abundance of larvae, should be able to add that as another predictor.
And John Waldman's question, did you compare your metrics of annual feeding success with the DEC annual juvenile abundance indices for shad and striped bass?
I think that's another version of the same question.
Same question.
I think that's another version of the same question, which is, can I use abundance of these early life stages of different species as a predictor in looking at diet composition and or feeding success? We can. I had Mike do that in what we call a fairly early stage of analysis when he was writing his thesis, but it's a subject that I need to return to.
Turn a slide back for Fran Dunwell, who wanted to see this slide. And is there any other questions in the room before I go on? Okay. This is another from Hoffman. I saw a zebra, this is just a comment that he saw a zebra mussel this summer in Sleepy Hollow during the DEC annual fish count. So If the Billion Oyster Project is very successful and the lower New York Bight waters clear, will that negatively affect feeding of various fish populations?
Wow, that's a great question. And I have some speculations, but they're merely only moderately informed speculations at this point. I think these pathways of production can be very complex. I think that as we've seen, zebra mussels themselves, the effect of zebra mussels may be fairly complicated because while they clearly have taken out one component of primary production, they may not have taken out all of them. I would imagine oysters, which of course are a natural part of the system, are going to have similarly complex effects. They are good at filtering water column. What do they filter? Do they filter phytoplankton? Do they filter bacteria? I don't know. I think that their selectivity may be fairly different from the zebra mussel. I see one member of my audience nodding in that regard. So the effect that they have is going to be, I would say, first, although not only, a function of exactly what particle size they're taking out of the water column. Then we'd want to know what it is they're adding to once they have taken that food and digested it. Bivalves produce these things called pseudofeces. So the result of their digestion, what is that contributing to the benthos? What is that contributing to the water column?
This was just a sort of a question related to oysters. And one of the interests that we've had in oyster restoration is the sort of three-dimensional habitat that oysters create. I'm wondering whether zebra mussels with, in certain areas do create that sort of three-dimensional structure, and are fish like shad attracted to those sort of structure, and are they capitalizing on that food source that the zebra mussels are taking in?
So my feeling about these early stage fishes is they primarily live in the water column, and they are not going to care too much about exactly what the bottom looks like. Now, it's pretty clear that once shad get to a particular size, they are feeding on the benthos. I know other river herring start to feed on the benthos once they get to a certain size. So they clearly start focusing in on the sorts of organisms that live in what we call the sort of epiphytes on the bottom, the aquatic vegetation that's on the bottom. And so at some stage, once they reach a certain size, they're going to start feeding on the bottom. And there's no doubt that the large amount of habitat that these zebra mussels create is going to have some change in the distribution of those feeding stages and the feeding opportunities that it affords them. But they're not going to be hanging out in that the way a demersal fish or a benthic fish would, because these early stage larvae of these species are pretty much in the water column.
I have a clarifying question.
Yeah.
I think you said that the co-spot and infobot data are all from a single spot, Kingston?
Kingston, yes.
And so if that's true, are you concerned that a lot of the correlations you showed wouldn't hold up if you had data, throughout the LS?
Well, fortunately, that Kingston site is within the region of the river that we were considering to be our upriver region, which is affected by zebra mussels. So in that sense, while it's not stratified spatially the same way that the longitudinal river survey is, I would say it's not unrepresentative, if you will, of what's happening in the mussel-impacted part of the river. Now, it certainly is true that we were using those predictors in our analysis of the downriver part of the analysis we did on striped bass. And the fact that we were still seeing a relationship between copepod abundance in Kingston and what's going on with feeding success downriver bears some head scratching, shall we say. And I'm not aware of sampling programs that would allow us to look at the extent to which there isn't a concerted along river pattern of zooplankton abundance in that way. I can tell you, and this is something I didn't mention, that there really, if you look at the upriver and downriver data sets, and you look at the feeding success variables, we did not, for the long-term feeding success variables, see any correlation in the interannual pattern of feeding success. So a good year upriver was not necessarily a good year downriver for long-term feeding success. I'll also slip in that short-term feeding success variable and long-term feeding success variable were intercorrelated, weakly but intercorrelated, meaning they were measuring the same thing just on slightly different time scales.
Oh, maybe just a quick follow-up.
Yeah.
The muscle density data,
Yeah.
that is river-wide, right?
So the zebra mussel filtration rate was again measured primarily based on measurements at Kingston and perhaps other places, I'm not sure, on mussel abundance and mussel biomass. So just the upriver portion, really, you have to view that as being the portion of the river, the portion of the estuary that the zebra mussels live in.
Thanks.
Yeah.
Justin Krebs says thank you to Hudson River Foundation and Dr. Schultz for a fantastic presentation. And have you been able to look at any diet samples for juvenile short-nosed or Atlantic sturgeon in terms of potential impacts of zebra mussels or other factors?
To my knowledge, we have very few sturgeon samples in the longitudinal river survey. So we have not analyzed that. We do have specimens of lots of other species and done some preliminary analyses of them. We know a little about tomcod. We know a little about rainbow smelt, remember them? But we have not done anything with sturgeon.
From Natalie Chung, are there any plans or have there been any attempts to manage the zebra mussel populations?
This is out of my area of expertise. Anybody?
I don't think there's been any attempt.
I know there's research on the possibilities. For instance, there was some investigation for introducing agents that would be selective molluscicides. I don't think there was a study of contraception as another possibility, but I think that the molluscicide project was started, but I don't think it ever amounted to anything.
And then except for cleaning the bottoms of boats and bringing more in, it's been more of a volcano watching exercise.
Yeah. And honestly, I mean, I think the way a lot of these invasions occur, it's a tendency for us to get very alarmed when they first occur because, of course, they're creating major changes in the system. And those changes are to some extent irreversible. However, we are seeing in the case of zebra mussels, and we're seeing that with many, although not all other invasions, that the pendulum swings back to some extent so that the effects become ameliorated or moderated.
I think in the Great Lakes that there was an invasion of the round goby following the zebrafish, and the round goby was finally now something that ate. Did I say zebrafish? Sorry.
No problem. You know what you meant.
And then the round goby brought down the zebra mussel population somewhat because it preyed on.
Right. So round goby had been introduced to the Great Lakes.
Not on purpose.
Not absolutely, and inadvertently introduced to the Great Lakes. And indeed, one of the marvelous attributes of this little fish is that it eats zebra mussels. I don't believe anybody has ever suggested that round goby ought to be introduced deliberately into the Hudson in order to further mitigate the effects of the zebra mussel invasion.
How about those new seaweeds that we're getting down in our area? Does that affect anything?
I would suspect it won't affect the zebra mussels just because I think we're talking about marine macrophytes that would not do well in the freshwater portion of the estuary.
We got a lot of that.
Okay? Indeed, we do in Long Island Sound.
I have a couple curiosities about the methodology, about the diet composition. Usually when you see a diet composition study, you see a pie chart.
Yes.
Where people show what
yes.
and the percent of each diet, and then they
yes.
have something which they were not able to quantify. So I was.
Oh, the unidentified part. Yes.
Is there something you have? I know it's very laborious technique to identify in such a small space. Do you have like a feel for what you have identified? Pilot represents the their diet, or another embedded question to that is, have you thought about using some proxies for the prey species like fatty acid signatures, which would shed more light on entire diet composition?
Okay, so there were two questions. One was the extent to which unidentified prey might have influenced our analysis of diet composition. Those diet composition numbers, the index that I mentioned to you, were based only on identified prey. So the extent to which there might have been temporal changes in unidentified prey is indeed interesting, and I have not actually analyzed that yet. That would be perhaps if there was a large component of unidentified prey that is sort of an amorphous mass, maybe in the shad case in particular, that might be, I don't know, zebra mussel larvae. So we need to look at that. The other question was whether we have other sorts of tracers of different prey components that we could use. I think that was your question. Do you mean like stable isotopes, for instance?
Stable isotopes or fatty acids?
Those certainly could be of interest. The stable isotopes, my understanding is stable isotopes have been looked at as a potential way of tracing the autochthonous production based on phytoplankton from bacterial production. But so far, to my understanding is there is nothing that's going to give one a particularly good read on whether, as I suggested, a lot of production might have shifted after the zebra mussel's arrival from a phytoplankton-based production to a bacterial-based production. But I'm still looking for that because I think that'd be a lovely project. Fatty acids are another way of tracing that. Now, unfortunately, certainly with respect to fatty acids, and probably as well with respect to stable isotopes, the samples that I've described here probably are not conducive to that kind of analysis because they've been kept in formalin, and so that long-term preservation in formaldehyde has probably disrupted the fatty acids. I'm getting no encouragement in that regard from the literature. But it's a great idea, so keep thinking. There might be something else there that we can use.
There was a follow-up question. I was hoping Judy would ask that question. One of the variables, which was missed in your study was the PCB contamination in the upper reaches of the river. And again, Judy would say more about it, but there's a lot of behavior effect of the contaminants on the feeding surface.
Yes.
Again, affect your analysis,
So if you have a variable, a predictor that I can use and employ in my regression analyses, please. If you think it's important, I'd love to test it. The striped bass manuscript has been accepted, so I'm not going to redo a lot of those analyses except for the sake of these presentations and starting and continuing a dialogue. But we're at a really good time for analyzing the shad data because I've just gone back into those data to prepare a manuscript for that. So
I don't know if they have data on, the level of PCB during, after, whatever, the cleanup.
Okay.
The Superfund cleanup up there in upriver.
Yes.
But, we have quite a bit of evidence that contaminants like PCBs and mercury will reduce feeding ability in juvenile fish.
All right, so a suggestion here would be, given that we saw two rather pronounced excursions downward in shad feeding success over the past decade and a half, shall we say, those of you that have some expertise in PCB mobilization might look at those and ask whether, as you suggest, perhaps that resuspension and mobilization of PCB might have negatively influenced. So,
I mean, if EPA or somebody has data on, you know, what was going on up there before and during the cleanup, dredging.
I missed it. What did you say the decline in shad was due to?
Well,
I'm only joking.
so, yeah.
Do you have any further thoughts on what next steps you might?
So I think, no, my understanding with shad, I think the ruling hypothesis right now is that there is there's a source of mortality acting on the adults that hasn't been well quantified nor managed. And that's an ocean intercept mortality. The story may be fairly similar for river herring, although the fishery is different. So there are fisheries that may be essentially over-harvesting these stocks relative to what they can support. I think that, you know, when you and I first talked about this project, you had suggested that there might be a problem with condition of the shad in the river. I can't speak to the condition of the adult shad. I can say that we're seeing some interesting events in the condition of the early stage larvae. And in particular, a couple years that really signify major changes in the condition of those larvae. And, you know, as I've said a couple times, semi-apologetically, I haven't finished analysis of these data, but I'm going to be very interested in whether those years have coincided with a sharp drop in the abundance of the larval stage relative to the juvenile stage. Because if those do represent years in which feeding success was very poor, we might expect that production of the juveniles was poor. I had Mike do an analysis of the feeding success relative to overall standing abundance of the life stage. So he said there's nothing there. But what he didn't do was actually look at the transition from the larval stage to the juvenile stage, which I think would be most important. So I'm going to look for a signal in whether those years represent times when, yeah, there might have been a lot of larvae around, but it just didn't amount to anything. Besides that, I don't see a long-term temporal decrease in the condition of these early stage fish. The early stage shad, admittedly, were doing very well before the zebra mussels arrived. There have been some years since they arrived, they were also doing pretty well. The juveniles don't show any real kind of temporal signature that would impress one as being at all related to the zebra mussels.
Any others? And one final question. Sarah.
Okay. One more from Sarah Hindle. Why do you think other species like the striped bass haven't taken advantage of zebra mussel vellers as a food source?
It's a great question. That's a great question. And I should say that given that I myself don't see, with the samples we have, representation of veligers in the diet, I can't say that stripers aren't feeding on them. I can only infer that shad are feeding on them because of this positive relationship between zebra mussel feeding rate and shad. But that, some may judge me as being way out on a limb with that one. It's certainly something that I would love to get more informed opinion on with respect to the extent to which those historic samples may have had zebra mussels in the diet. So I need to do more on that, for sure. So,
yes, I'd like to ask you a question about creating anchorages for oil tankers in that area. Would it change the ecology of your model?
Okay. Would anchorages for tankers affect the feeding ecology of fish? I'm not sure what features of an anchorage would possibly affect the feeding ecology. Do you have in mind any particular variable? Okay. Well, I mean, I think, you know, one thing about habitats that's important with respect to fish and early stage fishes is arrows that connect production upland to production in the river, and the arrows that connect different kinds of habitats in the river to one another. So benthic production is connected to pelagic production that's connected to fish feeding opportunities. If there are engineered structures that are reducing the amount of benthic production, I think that would have, to some extent, a negative effect on pelagic production, because that would represent a reduction in nutrients for things that happen on the bottom going back up into the water column. But that's a speculation.
Do we have any more questions online?
No more online.
No more online. Well, I think we've exhausted you here.
I'm still standing. Bring it, bring it. And thank you for your attention.
Okay,
Very
well,
much
thanks, everyone.
appreciated.
If you didn't sign in, we have a sign-in sheet outside at the desk. We'd like to keep track of you so we can inform you of future seminars. So thank you, everybody. Enjoy the holiday season. See you after the holidays.
The,